Welcome to the third lecture under immunology and immunobiology. This lecture focuses on clinical and diagnostic application of antigen antibody reaction. Also, the methods encompass anatomical techniques, immunological techniques, biochemical techniques, and serological techniques. And through these, we can identify specific antigens within tissue sections, cell proliferations in their complex and in unison through the following methods. These include immunohistochemistry, immunocytochemistry, and other serological techniques, which they are basically used in clinical and diagnostic application. Immunohistochemistry, as the terms imply, is a method used to detect the chemistry of immune complexes within the tissue, as the term histo indicates, histology, by viewing directly using transmission electron microscope under real time. Whereas the flow symmetry also detects immune complexes as they form as it react under staining technique by measuring the rate of diffraction of light, then transmitting it via an algorithm to produce a 3D structure of the complexes. Also, the serological techniques are the baseline techniques that we know in diagnosis of clinical infections and also detection of hematological status of an individual. These include detection of blood antigens, detection of the presence of other infectious viral diseases such as HIV, hepatitis, and other kind of infection. The immunohistochemistry techniques operate the way that ELISA is operated. The ELISA stands for the enzyme-linked immunosolvent assay. As you know, is an in vitro technique we use to detect the presence of an antigen or an antibody. However, in the case of the immunohistochemistry, this is something that is happening in vivo within the human body, and we can use this technique to detect its presence. As well, we can use it in vitro. It operates based on the principle of antigen-antibody binding. To identify an antigen, we can code that antigen with a fluorochrome, which can give us an emitted color to indicate whether there is a reaction between the antigen and the antibody. Most especially, it is used to identify specific antigen protein within the tissue by using a level antibody as I've said. And conventionally, it is used in diagnosing several types of cancers. The most immediate example is the breast cancer cells. We can use it in mapping the distribution of immunoglobulins in autoimmune disease, which we are going to discuss in detail in the coming classes. So the principle of operation of the immunohistochemistry revolves around visualizing antigen-antibody reaction in real time within the tissue, as I've said. But the most important component of this technique is that it possesses what is called a reporter, a horseradish peroxidase enzyme. This is used to tag an antibody that is binding to the antigen to detect the presence of that antibody. And it is bounded by a color precipitin, or you can call it a fluorochrome, which emit a light as a substrate whenever there is that reaction. So the baseline is that the principle is operating exactly like a sandwich ELISA technique, if you can recall from your undergraduate classes. So it produces a color when there is the reaction, and it can be tagged directly using a fluorochrome if it is a direct immunohistochemistry, and if it is a sandwich, you can use a secondary antibody labeled by the horseradish, which is tagged by the fluorochrome that usually emit light in the presence of the substrate. First step for immunohistochemistry technique is tissue proliferation. First of all, this proliferation technique follows 5 distinct stages. The first stage is fixation, where the excised tissue or sample tissue will be immersed into a concentration of 4% formaldehyde, paraformaldehyde, and incubated at 24 hours, usually at 4 degrees Celsius. Then it will be dehydrated to remove excess water. It will move to clear it, using xylene at first concentration and second concentration. The variable concentration will give you a chance to determine which one is most fit for the clearing. Then you move forward to paraffin infiltration. This is just used to remove some certain component from it and make it relatively hard. It is dissolved in paraffin under water bath at 56 to 58 degrees Celsius. From there, the tissue will be embedded in a particular machine which will slice into smaller pieces, small pieces that can be fitted on a microscopic slide for easier microscopy. The second most important aspect of the immunohistochemistry technique is that after tissue preparation, we also have to remove the paraffin and rehydrate. A reverse of the process exactly happened during the tissue treatment. We consolidated and solidified the tissue using paraffin, and therefore to see the exact reaction, we have to remove the paraffin. So we reverse it by treating it again with xylene. So the slide will be deposited inside the xylene and will be treated for just 3 to 10 minutes. Changes will change because the paraffin will be removed entirely. Then to remove the water, I'm going to treat it with ethanol at 100%. At 100% if you want to remove the water from the processed tissue, but you can vary it so that your result can be analyzed by different concentration of the ethanol treated during the technique. In the third stage of the procedure, we have to recall that the fixation, paraffination, and deparaffination can mask the antigen on the surface of the tissue. So because of that, we have to revive the confirmatory part of the antigen on the surface of the tissue, and this can be done by treating it first of all more traditionally with heat inducement so that we can retrieve the epitope, that's the binding site on the surface of the antigen. Another method that we can also use is the use of enzyme, proteolytic enzyme, which reshaped or degraded the protein aspect of the antigen in question. So we use this to revive the structural conformation of the antigen or the antibody by just breaking the protein components superficially on the surface of the antigen or the antibody. To prevent the excess blocking of serum or protein added in the case of the antigen, we employ a process called deblocking, which we usually use the horseradish protein or the alkaline phosphatase. In the case, this also varies with the kind of tissue you are treating. In the case of either liver or the spleen, we use the horseradish protein, whereas if it is the tissue is often from the intestine or the kidney, we use the alkaline phosphatase. This will remove all the excess protein so that it will prevent non-specific binding between the antigen in question and the antibody. Now, after blocking, we've already removed all the residual aspects that may bind to the antigen in question. So the next stage is we are going to add the primary antibody, which is technically used to detect the presence of the antigen. As you know, the reaction between the antigen and antibody is extremely specific. So where there is any complex form after the addition of the primary antibody, it is indicating that yes, there is a reaction, and yes, that the antigen in question is the antigen that is expected since there is a binding between the primary antibody. So the slide is incubated with the antibody specifically to the target protein, usually 1 hour at 4 degrees Celsius. Then it can be washed with PBS buff. At the sixth stage, since it is a sandwich process, the preferred tagged antibody, which will bind to the primary antibody, is an antibody that is tagged with a substrate and a reporter protein, which is the horseradish protein. So where the horseradish tagged antibody bind to the primary antibody, a substrate would be added. That substrate, when added, it will react with the primary antibody, which is just tagged with the chromogen, as I've said, to emit a light. So that light is showing you that yes, there is a reaction. Because technically, the reaction between the primary and antibody and the antigen cannot be vividly seen. However, when you tagged it with a secondary antibody and reacted with the substrate, you are going to see a color change. That color change is indicating that yes, there is a reaction between the test antigen and the primary antibody. Now, the seventh stage, which will be counter staining, the entire complex will be stained using hematoxicillin, and it will be washed immediately. Then it will be air dried. After air drying, a cover slide will be added. As you know, when you want to conduct a direct staining using a farmland slide, you have to dry it first. Then it can be viewed in electronic microscope. So all the color and the complexes can be seen vividly using a microscope under a cover slide. So at the end, the morphology, the structure of the complexes on the surface of the tissue can be vividly seen via a monitor. The immunohistochemistry technique has a very vast application in the medical aspect, especially in clinical diagnostics that has to do with infectious agent. So first of all, cancer diagnosis. There are a lot of other types of cancer that involves the tissue and the immune cells and organs. So we can use this in detection of carcinoma, melanoma, and lymphoma. Another thing is that in the determination of the primary site of a tumor or a cancer, there is a localized cancer where it affects one tissue, and there is a situation where a cancer it can progress and metastasize to several parts of the body. So we can use the immunohistochemistry markers to detect the original part where the tumor started. Another most important application of immunohistochemistry is that certain pathogens from viral and bacterial antigens can be detected directly on the tissue. This is also directly applied to the procedure as we have explained it. So in the case of, let's say, the Helicobacter pylori, which is a pathogen that is causing a secondary ulceration in the human body, that can be used to detect the presence of this etiological agent on the surface of the stomach. Another technique used in clinical and diagnostics approach under immunology is flow cytometry. The flow cytometry analysis measures physical and chemical characteristics of cell and tissue whenever there is a immune cell reaction. However, it is widely used in the area of immunology, microbiology, hematology, and clinical diagnosis. It is operating based on the principle of binding antigen with a fluorescent antibody. The principle of operation of the flow cytometry depends on antigen-antibody reaction as well. It is also related to the immune histochemistry technique, where the antibodies will be coated with a fluorescent dye so that the reaction will be vividly seen. However, in this case, the color are not just physically seen, they are noticed by passing a laser beam forward scatter in such a way that it can measure the size of the cell, and also such scatter in such a way that the granules and the complexity of the cell can be noticed. So the granularity within the cell depends on the cell component within the cytoplasm of the cell. So this can be detected using a laser beam, and a signal identified will be determined based on the algorithm in the computer system. Now, the main component of the flow cytometry or cytometer composed of four components: the fluidic system, where the preferred cells are transported and they will be sorted out one by one so that the laser can pass through. Then there is the optical system, where the diffracted light will be collected into segment. Then there is the electronic system, where this diffracted light will be amplified. Then there is also the computer system, where it will be transcribed into an image so that it can be vividly seen. The first basic procedure for flow cytometry is sample preparation, and this sample can be biological sample, which can be either blood sample, tissue sample, bone marrow, cells, and other things. And these are prepared first of all by centrifugation. This centrifugation can be between 5,000 to 20,000 revolution per minute, depending on the type of sample we are processing. Then the cells will be counted based on the volume of cell required to pass through the cytometer. Then on the plates, it will be blocked to make sure that there is no unspecific binding between the antigen and the antibody on the surface of the cell. Then it will be tagged or stamped using the tagged antibody, which we are using in noticing the rate of the reaction, using a fluorochrome, a dye content. It will be washed using a soluble, suitable buffer, and the complexity of the sample can be noticed using a computer. If that is done, then we'll move forward to the real procedure. The amount of cell will be injected into a cytometer. Then it will be agitated in such a way that one cell will pass through the laser beam at a time. So if it is passed through the laser beam, it can be either a forward laser beam in such a way that it can measure the size of the cell, and also a side laser beam where it will measure the complexity and granulation of the cell. This is trying to say that the organelles within the cell will be also detected and measured, as well the protein substances or on the surface of the cell can be detected. In the case of you can see CD complex, the cluster of differentiations, which we have a lot of subset. Then the scattered light will be collected, by of the scattered light will be collected together. Then an algorithm will interpret it using a specific software, where we can see the 2D composition of the organelles on the surface of the cell and within the cell, and we can also notice the size of the cell. based on the diffracted light, scattered light collected from the laser beam. So from there, we can notice a lot of things. So as we know that the flow cytometry cut across a lot of professions, from immunology, microbiology, cytology, and a lot of other professions. However, in the case of immunology, it also has a vast application. First of all, in immunophenotyping of immune cells, we have a lot of immune cells that we can detect its morphology and its application using flow cytometry and the surface proteins on the surface of those immune cells. We can also use in detecting the cluster of differentiation markers on lymphocytes. This can be either B cell or T cell. There are a lot of cluster of differentiation. We have cluster of differentiation 4, 13, 19, 20. There are a lot. We can analyze T cell, B cell, and natural killer cell. This is exactly the same with immunophenotyping. We can also measure the percentage or concentration of cytokines production and their action. As you know, they are working based on concentration gradient. So we can know how much concentration they are in a localized area where there is an immune reaction. Baseline here is that there are a lot of other applications that we can employ during research. This includes to detect a cell cycle of each and every immune cell or other blood cells within the human body. We can also measure the mechanism of apoptosis of a cell in the case where there is the action of macrophages or natural killer cells. We can also measure the cell proliferation studies, how cells are produced, how they are multiplied, and how they are disseminated to different parts of the body. It is also employed in stem cell technology, the production of the stem cells and how it can differentiate into so many other cells within the human body. Then we can also use in sorting, production of vaccine, and its relationship with drug development. So it's a very important procedure which immunologists and microbiologists cannot entirely dodge, especially in the case of an MSc study. Another most important technique we use in clinical diagnosis is serological techniques. This is also applicable in microbiology, immunology, and hematology. Serology is just a study of the serum and other bodily fluids and how they are related with immune cells and other antigens. Clinicians use the concentration of antibodies in the blood. As well, it is essential for tracking the progression of infection like hepatitis, COVID-19, rubella, HIV, and a lot of other diseases. By looking at how concentration of these antibodies rise and fall over time, doctors can determine if an infection is either a new infection by the presence and concentration of IgM, which you know it is the first immunoglobulin that is produced in the advent of an availability of foreign substances within the body. And you know that the IgM is a pentamer, is a very large immunoglobulin and very effective. As well, if you notice the availability of IgG, which is a monomer, in the blood circulation, it is indicating that the infection, it is an old infection, it is convalescent. So that's why the measurement of these immunoglobulins in the serum is very effective. As well, if a vaccine has provided sufficient immunity, it's something that we can also use in using serological analysis. So serology is extremely essential in clinical diagnosis of infectious agent and even other systemic infection in the case of autoimmune diseases. Serologically, we can measure the rate of reaction between the antigen and antibody in vitro outside the human body. Very straightforward, we are just mimicking what is happening within the human body, outside the human body, so that we can clearly diagnose and treat infections outside the human body. So following this serological analysis, there are techniques or methods we employ that aided us in diagnosis of infections related to immunology and hematology. And this method include enzyme-linked immunosolvent assay, the agglutination test, complement fixation assay, neutralization test, and opsonization reaction. These are very essential in immunology, as I've said, and we use them in determining the presence of infection in the human body and how we can treat it. ELISA is a biochemical technique used to detect the presence of antigen or an antibody in a sample. However, the principle of operation of this technique is detecting the presence of this reaction using a signal tag, as we explained. So the baseline for the principle is almost the same with the immunohistochemistry flow cytometry, especially in the part where we are purifying the sample. However, this doesn't require any additional laser beam or microscopy to detect the rate of the binding or the presence of the binding between the antigen and the antibody. However, the fluorochrome dye in the presence of the substrate can emit a light which can clearly see if the reaction is there and it is available. So that's why ELISA is very straightforward and it is rapid. So it's not time consuming and is effective in diagnosis of clinical infections. So the procedure is also straightforward. First of all, we are usually using a microplate, or some people they call it the 96 plate, where the plate will be coated with the antigen, the test antigen or the test antibody, depending on what you are trying to check. Is it the antibody or the antigen? Then the non-specific or non-reactive part within the microplate will be blocked. So a non-reactive protein, most often the bovine serum or albumin, is added to cover any remaining sticky spot on the surface of the microplate to make sure that there is non-specific binding after the antigen or the antibody is added on the test antigen or test antibody. Then the sample, which can be either a serum or a plasma, will be added. Then it will be thoroughly washed to remove excessive test antigen or antibody. The plate will be rinsed to remove any unbound molecules, can be either protein or any substances. Then to detect it, an enzyme link antibody is added, followed by another wash. So the enzyme link antibody, if you can see from here in the step 3, the enzyme label detected antibody is just an antibody that you add it, which will bind to the fluorochrome and the substrate so that there will be emission of light. So after you add it, you remove the excess by washing again. Then since it is already tagged with the fluorochrome, then the substrate will be added. So the substrate will react with the fluorochrome to emit the light. So the availability of the fluorochrome and the substrate will produce a light, but the intensity of the light will depend on the rate of reaction between the antigen and the antibody. So there are 3 to 4 types of ELISA. There is a competitive ELISA, where first of all the antigen will be binded with the secondary antibody with the substrate. Then another secondary antibody will be added. So this is a competitive ELISA. So it is depending on which the types of light emitted depend on which type of antibody binded to the antigen. Then we have a sandwich ELISA. Sandwich ELISA, from the name, is just an ELISA technique where it's just an arrangement of a sandwich. So there is first of all an antibody from the below, then you add your antigen. When you add your antigen, you add another antibody which will react to the other antibody that you add. So which is a tagged antibody with the fluorochrome that will react to emit the light for you. So that's why it is called a sandwich. There is antibody below and there is another antibody above. The antigen is at the middle. So that's why it is called a sandwich. Then we have an indirect ELISA where the antigen is at the below. Then you add your secondary measurement antibody. Then the tagged antibody will be added. So it is indirect. The tagged antibody is not binded to the antigen. It is binded to another antibody. Then we have a direct ELISA where the antigen will be binded together with the tagged antibody and the substrate. This is very straightforward. It is not complicated and it's very straightforward. So whenever there is a reaction, you will just notice it directly immediately after you add your test antibody. Then you add your substrate, you see light will be emitted. So this depends on the stages, the type of sample you are dealing with, and the rate of the reaction you are expecting between the antigen and the antibody. So it's effective and very straightforward, and it has a very vast application. Now, agglutination reaction. If you happen to see how the kind of reaction that is taking place when you are testing, or you want to detect the presence of a typhoid fever using Widal test, or you want to detect the type of blood group using blood grouping analysis. So the clumping that you are seeing, it is called agglutination. That is because there is a reaction or clumping between the antigen and the antibody. So the principle is revolving around the valency and several types of epitope on the surface of the antibody. As you know, the IgG is a monovalent. It is one immunoglobulin with 2 binding sites at the variable site. Whereas if it is immunoglobulin M, you know it is a pentamer. It has a variety of binding site. So where there is a clumping between the epitope and the paratope, it creates a linking bridge between the antigen and the antibody. So you will see that the blood will be agglutinated, if you can recall your Wadel test and the blood group test. And this can vary. If it is between viruses and red blood cell, the clumping, you see that it is not localized. It is a dispersed clumping on the surface of your slide. But in the case where there is an antibody and a virus, a red blood cell, you see that it is a generalized clumping on the surface of the slide. So that's why agglutination is very straightforward. It is a qualitative reaction where you can just measure by visualizing. So it's a very straightforward. We can use it in detecting the presence of blood grouping, in detecting the presence of H. pylori, hepatitis, and other type of infection. So it's a very good method employed in diagnosis of clinical infection. So there are two types of agglutination tests, and they comes together with their procedure. The slide test, I know most students they are conversant with it, where you add your sample together with the antigen. So you rock it within 1 to 2 minutes, and you see whether there is agglutination or there is no agglutination. So it's a qualitative procedure, and it's very straightforward. That's why example of them is the presence of salmonella in the blood, where we tested Widal. And in the case of identification of Staphylococcus aureus, if you can recall, we are using hydrogen peroxide. So the reaction between Staphylococcus aureus and hydrogen peroxide breaks down hydrogen peroxide into water and oxygen. So if it did that, you see that there is a clumping. That clumping is indicating that there is an enzyme which breaks down hydrogen peroxide into water and oxygen. So it's indicating an agglutination reaction. So there is also tube agglutination procedure, which is quantitative in nature. Usually start with a serial dilution to make sure that there is a variability of concentration. Then you add your antigen. So the presence of the antigen will determine the rate of agglutination, or the quality of the quantity of the agglutination at variable concentration. So that's why agglutination reaction can come either to measure the qualitative nature of the reaction and also the quantitative nature of the reaction. So it's very important also in diagnosis of infection, and also for detection, for phenotypic detection of microorganisms, as we earlier explained in the case of Staphylococcus aureus. To contribute to this very lecture and to measure your understanding of the course, this is an assignment for you. This is a general assignment, so everyone will write on this. You are going to write on the principle, the procedure, and application of each of the following. One is opsonization reaction, which you can know is antigen presentation. Then there is complement fixation, then neutralization reaction, which works based on the valency between the antigen and antibody reaction. So I want you to do this assignment. I will share a link for you where you submit the assignment. Then there will be another link for MCQ where you drop your attendance and answer the questions. This is important. This is a general assignment for everybody. Gradually, we are going to allocate topic for everyone in the class so that you come and make a presentation. A very good aspect of MSc is you have to be conversant with making presentation, sourcing of data by yourself, and reading the data by yourself. This contributed to production, or should I say creation of knowledge. We don't have to continue utilizing knowledge. We have to give students chance to add their idea, so that by adding your idea and opinion, you're also creating knowledge. So that's why the assignment is very essential. So use this generalized assignment, submit it. As I've said, I'll indicate a time frame for you to make the submission and to answer the MCQ. So thank you. Where there is a question, you can drop it to me via private message. And I think this will be our last online class. And after we resume, we are going to start with physical class in the university. So thank you for your time.